Hello, everyone. Good morning, good afternoon, and good evening. Marcus Segribi here, Managing Director from MSQ Ventures. We are honored today to host Mr. Gino Germano, President and Chief Executive Officer of Elucida Oncology, to our MSQ CEO webinar series. Elucida is a clinical stage biotech company with its targeted cancer therapy, first in class, ultra small nanoparticle CDOT drug conjugate CDC platform. CDCs penetrate deeper into tumors and deliver significantly higher payload versus ADCs. They recently announced first patient dosed with their lead asset. And congratulations on your recent fundraising rounds. Mr. Gino Germano, how are you today?
I'm doing well. How are you?
Good, good, good. Enjoying some beautiful weather here in New York. Where are you calling from?
I'm just a little bit south. We're down in the Philadelphia area.
Okay, the Northeast area. Great, excellent. We have our audience is global in nature, so spanning 20 plus countries. We have a strong presence in China, so good evening, Shanghai. Jumping into the webinar interview here, really would love to learn a little more about your background of your journey. So prior to joining Elucida Oncology, Mr. Gino Germano was president of Intrexon, held multiple executive level roles at Pfizer, J&J, Wyeth. So Mr. Germano, please share a little of your inspirational journey over the past few decades, which led you to Elucida today.
Yeah, sure. Happy to. Well, I really feel blessed to have found the pharmaceutical industry and the life sciences field. I've been in the industry for over 35 years. As you mentioned, I began my career with Wyeth Pharmaceuticals. I was there for about 25 years. And I spent a few years at Johnson & Johnson, and then ultimately the last portion of my career was at Pfizer. I've had the good fortune of being able to work on some really, life-changing and life-improving technologies over the years, and it's incredibly enriching experience to do this type of work, and it's always been extremely motivational for me. I retired from Pfizer. I was the group president of the innovative pharmaceutical business there, which was a pretty big job, and had a lot of experiences across many different therapeutic areas in all parts of the world. I managed a development organization that did drug development. I co-chaired our research strategy council. So I was really involved with, research, decision-making, portfolio prioritization, drug development, and global commercialization of a very broad portfolio of products. I retired in 2016 and spent the first couple of years doing some board work and some consulting. And I spent a short period of time at Intrexon doing some gene editing work there. And then I became acquainted with Elucida. And, it was actually Bob Esner, who was formerly chairman and CEO of Wyeth Pharmaceuticals, who contacted me and introduced me to the founders of Elucida. And as I grew to learn about the technology, I just got more and more excited about the potential for this platform to really take the treatment of cancer to the next level, and across a broad array of different opportunities, which was also exciting. And then the idea of really building a company almost from scratch. There were two or three employees. I think it was the third employee. So it all kind of motivated me, and I started down the journey about 5 years ago, and we figured out what to do with the technology and started developing our first candidate. And now, as you mentioned, we're in the clinic. So it's been an exciting experience so far.
Wow, amazing! I'm sure, looking forward to learning more about that journey as you present in a few. But just I guess more so on the company vision. Elucida is pioneering with its ultra small nanoparticle CDOT drug conjugate platform, CDC. Yeah. So how do you envision this platform evolving in the next 5 to 10 years? What kind of impact do you anticipate it will have on the broader oncology landscape. I know you're going to talk about it in your presentation, but maybe high level.
Sure, sure. So, actually, back when I was at Wyeth, maybe a decade ago, or it could have been 2 decades ago, I actually had the good fortune to launch the very first antibody drug conjugate that was registered in the United States, a drug called Mylotarg, which subsequently was launched on a global basis. And I was pretty enamored with the opportunity to work with a new technology and this whole concept of targeting cancer cells and delivering drug payload directly to the cancer cells with the goal of trying to avoid off-target toxicities. That just seemed like it made an awful lot of sense to me. And so, I started working with the antibody drug conjugate class and realized that there were some certain advantages over chemotherapeutic agents as just standard agents when you can attach them to an antibody and deliver them directly to the tumor. But we also saw that there were some shortcomings of the antibody drug conjugates as well. There were some limitations to how well they worked, and some issues to still overcome. So when I became aware of this CDOT technology, it really did have the ability to do what antibody drug conjugates do, target cancer cells and deliver a drug payload. But it had some advantages in that would overcome some of the limitations of the antibody drug conjugates. For example, antibodies tend not to be able to penetrate into solid tumors. They kind of adhere to the surface, whereas our particles are actually able to penetrate deeply into solid tumors and engage with more tumor cells initially. We can carry substantially more drug payload, like 10 times more. So we can get a lot more drug to a lot more cancer cells more quickly. And then the third thing is the particles tend not to interact with healthy tissues in the body. They circulate in the bloodstream, they leak into tumors where they're retained, and they avoid uptake into healthy tissues. So they do indeed reduce off-target toxicity. So I just thought this was an ideal technology and can be applied very, very broadly in the field of oncology. We can use almost any small molecule drug. We can even deliver radionuclides to tumor cells for targeted radiotherapy. So, I see a future where this technology can be leveraged across a broad array of different tumor targets, as well as different drug payloads. Because we can put so many more molecules of drug on each particle, we're not restricted to just highly potent cytotoxic drugs. We could use other cancer drugs and enhance their effectiveness by getting more drug to the tumor and reducing the off-target exposure. So I see a big future for Elucida and our ability to develop our compounds in-house. And I also see a huge opportunity to partner with strategics across the world to develop CDOT drug conjugates for them using proprietary payloads or targets that they may be interested in. Really, the sky is the limit with this technology.
Exciting. Really looking forward to the future here with you and the progress. Recently, you raised a round. Capital markets have been tough. Congratulations, and please share any insights that you have in terms of what just generally, I guess, in terms of the biopharma capital markets outlook from your perspective for the rest of the year going to 2024. What kind of advice can you give some of the audience members?
Yeah, I would say 2022 and into the early parts of this year were pretty challenging, especially for privates, and we're still private. Especially with institutional investors, there's very little activity for a while there. It's starting to warm up a little bit now. Our raise we essentially went back to insiders, and they've been very excited about the progress that we've made. So it was not that difficult to raise some additional funds from them. But I am looking ahead, and in this business, you have to constantly pay attention to the cash balance and your runway, and I would like to see us continue to develop our clinical data set that can be hopefully will be as compelling as the preclinical data that we generated previously. I think we're on our way, and I'll share some of that information in my presentation. But I hope that we continue to see the markets improve. and see a good flow of cash into exciting companies like Elucida. We expect that we'll be looking at the beginning of next year as our data set matures a little bit to go back to the markets for a B round or a crossover as we prepare for an eventual IPO. So, I'm optimistic, and I'm seeing already some signs of improvement, and I hope that continues.
Great. Yes, we're looking to really, I guess, exit this winter.
Yeah.
Overhanging us, looking for those green shoots. So whenever we see success capital raises like this, either in the private or public markets or follow-ons, we're always paying attention. So thank you for your insights. Now we look forward to your knowledge sharing of Elucida Oncology's novel approach to its first-in-class targeted cancer therapy using your novel and differentiated CDC platform. We'll take questions from the audience on the back of your presentations. So please, Mr. Gino Germano, please share your screen and start your company presentation.
That sounds great. Let me just pull it up here. Okay. So as we've been discussing, the technology that Elucida is founded on is called a CDOT. It was invented at Cornell University. That's why it's called a CDOT. And we licensed the IP from Cornell and Memorial Sloan Kettering Cancer Center, who did a lot of the early translational work. So we make CDOT drug conjugates. As I mentioned before, they target cancer cells and deliver drug payload like antibody drug conjugates. But the advantages that we bring are the ability to actually penetrate deeply into the tumors, deliver substantially more drug payload, and then avoid uptake into healthy tissues and clear from the bloodstream through renal excretion. So we call this target or clear. They either target the cancer or they clear from the body. And I'll show you some of the results of that. Our lead program is called ELU-001. It is a folate receptor alpha targeted CDC, and this is a target that's seen a lot of excitement in the recent months, and we're looking forward to advancing our program. We have data now on about 30 patients. We've seen some RECIST responses across a variety of different tumor types, and importantly, across a wide range of antigen expression levels. So in many cases, antibody drug conjugates require fairly high levels of antigen expression for activity. We're seeing activity across high, moderate, and low levels of antigen expression, which is really good. We've also seen a clearly differentiated safety profile. We're not seeing any of the off-target toxicities that are common with ADCs, like peripheral neuropathy, interstitial lung disease, cardiac, liver, kidney, ocular toxicities. We don't see any of those toxicities in our studies to date. And we're now advancing into dose expansion cohorts in ovarian and endometrial cancer. They started already this year. We're enrolling patients, and we plan to test our compound in actually a liquid tumor as well later this year, a pediatric form of AML that also expresses folate receptor alpha. We have an exceptional team that I'll introduce you to in just a moment, and multiple inflection points as we go through this year. And this is kind of a picture of our portfolio. On the top is ELU-001, the lead compound, the folate receptor alpha targeted CDC. And you can see the solid tumor program is now in the clinic. The liquid tumor program will be in the clinic soon. Another differentiating feature of our technology is its ability to deliver drug payloads to difficult to reach tumors, including brain metastases and primary glioma. We have quite a robust set of preclinical data demonstrating the capability of the technology to achieve this goal, and we're looking forward to advancing a program in the brain metastasis field in the near future. We have a research collaboration underway with a major pharma company that was interested in our technology. Actually, they're one of the top players in the ADC field, and they recognize the potential benefits of our technology. So we're working with them to develop a CDOT drug conjugate with a target that they're interested in, and a drug payload that they would like to use. And then we have additional compounds in earlier stages of development. So to kind of characterize our vision, we have a small particle with a really big potential. So far, we've been talking about CDCs or CDOT drug conjugates. So this is where we take the particle and we functionalize it with a targeting molecule or a binder, and then we add drug payloads through a cleavable linker. And we have the ability to utilize this technology with almost any binder and almost any small molecule drugs. When you think about how many targets are known in cancer, there's got to be 40 or 50 that have already been validated, and then any number of small molecule drugs, you can kind of mix and match those components and come up with an incredibly robust portfolio of compounds. But it doesn't stop there. We also have the ability to develop what we call radio CDOT therapeutics by attaching a radioisotope to the particle instead of the drug. And then we can deliver the isotope to the tumor. And if it's a alpha emitter or beta emitter, we can achieve targeted radiotherapy. And because of the penetrating capabilities of the particle, we can get that radiotherapy directly to many tumor cells in a short period of time. And then finally, we can play in the IO field or the immuno-oncology field by delivering molecules that are immunostimulatory and potentially synergistic with other immuno-oncology compounds like Keytruda or other PD-1 antibody agents. To go into a little bit more detail on exactly what a CDC is, it starts with the synthesis of a ultra small silica spheroid. You can see the scale there. The spheroid is about 4 nanometers in diameter. So most of the time when you hear about nanotechnology, you're hearing about something that is 100, 200, 300 nanometers in size. This is 4 nanometers in size, and it's that ultra small size that enables that tumor penetration and renal clearance that offer several benefits to the patients. So silica is a highly biocompatible material, so it's ideal for this use. And once we have made the particles, we attach short-chain polyethylene glycol molecules to the silica. And the pegs serve 2 purposes. One, they render the particle neutral in charge, so it avoids interaction with proteins and other charged molecules in the body. And this also perhaps contributes to the tumor penetrating capabilities of our technology. The pegs also serve as the anchor point for the attachment of the targeting ligands or binders. These are the molecules that engage with tumor cell surface antigens. And then we add the drug payload through a cleavable linker. And just as I've shown you in this illustration, we kind of build this product and we have exquisite control over how we do that. So if we want to build a product that has 5 targeting ligands and 10 payloads, we can do that. If we want 10 targeting ligands and 20 payloads, we can do that. So we have the ability to actually iterate in the development process and optimize our product before we go into clinical development. And what are our goals? Well, our goals are to achieve clear differentiation from the antibody drug conjugate or other conjugate products in positive ways. So we want to create CDOT drug conjugates that are effective against high antigen expressing cancer cells, but also are effective against moderate and low antigen expressing cancer cells, so that we can address a larger patient population with different tumor characteristics. We test for deep penetration into solid tumors, as well as retention within those tumors for a long-term effect. We want our CDCs to cross the disrupted blood-brain barrier that's associated with brain metastases, so that we can address the brain metastases that often occur with tumor types like lung cancer and breast cancer, for example. We're looking for rapid tumor uptake, but with a relatively short circulating half-life. We will deliver significantly more drug payload and avoid uptake into the liver or the reticuloendothelial system. And we have assays to evaluate our compounds across all of these parameters. So let me show you a little bit of data. This data was generated at Memorial Sloan Kettering early on, and this is kind of what attracted me to the company in the first place. What we see here on the left is when we administer the CDC to animals, we see the uptake at the site of the tumor. That's the red bar. It's a rapid uptake, and you see this is a single administration, and there's long-term retention within the tumor. So the particles get in there and they stay there. So they have time to interact with tumor cells and release payload and effectively treat that tumor. But if you look at the bottom graphic, you can see the clearance from the bloodstream in these animals also occurs fairly rapidly. So after just a couple of days, 48, 50 hours, most of the drug is out of the bloodstream. But if you look at the tumor, there's still therapeutic levels of drug at the tumor. So this is a completely different paradigm than what you'd see with an antibody drug conjugate, where you need to have continuous blood levels of the drug in order to continue to replenish the drug at the site of the tumor. This is really ideal for this type of therapy. And on the right-hand side, you see the same thing in a human study, where after administration of the CDC, you see uptake at the site of the lesion in the head. This is a pituitary adenocarcinoma. So you see that the CDCs are getting to the tumor and staying there. And when you look at the full body scan, you see that the residual drug is accumulating in the bladder for elimination from the body. So we call this, again, target or clear. And this results in a very favorable biodistribution pattern, where when we look at tissues and organs of interest, you can see that after administration of the CDOT drug conjugate, there's initial exposure because the CDOTs are in the blood pool circulating through these tissues, but you don't see any retention like you saw in the tumor in the previous slide. So in all of these organ and tissue systems, they really don't experience contact with the CDOT drug conjugates in any appreciable way. So just to show you a side-by-side comparison of ELU-001, our folate receptor alpha targeted CDC, compared with folate receptor alpha targeted ADCs. ImmunoGen has one actually on the market. It's called Elahere, and Sutro is working on developing their own folate receptor alpha targeted ADC. So what are the differences between Elucida's compound and the ADCs in development? Well, it starts with the payload. We've chosen a different payload, a topoisomerase 1 inhibitor called exatecan, very similar to the payload that's used in Daiichi Sankyo's Enhertu. We chose this because we believe it's more active than the tubulin inhibitors that are associated with the antibody drug conjugates on this chart. Topoisomerase 1 inhibitors are effective in both dividing and non-dividing cells, whereas the tubulin inhibitors generally require active cell division in order to have activity. So we first chose a more active drug payload, and then we put a lot more payload on each particle. So the DAR is drug antibody ratio, or in our case, drug particle ratio. So we have 21 molecules of drug per particle, whereas the antibody drug conjugates carry 3 to 4 molecules of drug. We also have more binding sites or binding molecules. So we have about 13 per particle versus the 2 that you see with an antibody has 2 binding sites. Because we have more binding sites, we believe that this is responsible for greater avidity and ability to target lower antigen expressing tumor cells. Next is our tumor penetrating capabilities, and I've mentioned that before. This is a property that we have, and the antibody drug conjugates generally do not. And then in terms of systemic exposure, we have a shorter circulating half-life, so there is reduced systemic exposure. So quite a different paradigm. And these are the data that show that tumor penetrating capability. So these images are slices deeper and deeper into a tumor spheroid. And on the top, you see tumors that were exposed to ELU-001. And as you look at slices deeper and deeper into the tumor, you see the fluorescence remains. The drug is fluorescently tagged. So where you see fluorescence, you know there's drug there. On the bottom series is the antibody drug conjugate, mirvetuximab. And you can see, as expected, the ADC is adhering to the surface of the spheroids, but not penetrating inside the spheroid. And these are data that demonstrate a side-by-side comparison of our CDC to a ADC that is a folate receptor targeted ADC fashioned after mirvetuximab or ImmunoGen's ADC. And what we have here is different tumor spheroids with different tumors and different levels of antigen expression. So in the parentheses under the bars is the amount of expression. So a 1+ is a low expressing tumor, a 2+ is a moderate expressing tumor, and a 3+ is a high expressing tumor. And you can see kind of across the board, 27 or 28 different tumor types here. We're comparing the effectiveness of ELU-001 or the potency of ELU-001 versus the antibody drug conjugate. So the less drug you need to kill these tumors, the more potent you are. So in the violet or red color is the CDC, and you can see in almost every case the CDC is more active than the ADC, regardless of the origin of the tumor or the level of expression. And we saw this in vivo as well. On the left, you see a comparison of ELU-001 in the red or pink color compared with the ADC in the gray diamonds. The ADC did have some activity, but not nearly as much as the CDC. This is when the drug was administered every 3 days times 3, and then discontinued. And then on the right-hand side, you see that we can actually administer it less frequently and still get the same effect. So this is administered every 7 days times 3. And the reason we can administer it less frequently is because the particles get into the tumor and are retained there. You don't have to administer quite as frequently. These are data demonstrating the effectiveness of ELU-001 in a brain metastasis model, and the images show you that the drug is getting into the area of the brain where the tumor exists, but not into the healthy part of the brain. And the graphics on the right, on the top, you can see that when ELU-001 is administered to the animals that have these metastases, it's having anti-tumor activity when administered when the tumors are small, as well as when they get larger. And this is data from the pediatric AML model in animals. And this is really remarkable data because you can see in the images that the patient, or the animals that were treated with normal saline, had obviously progression of their cancer, and by day 56, all of the animals were dead. In contrast, when ELU-001 was administered to these animals with one course of therapy, 3 doses, the cancer was eliminated, and we had complete responses in all animals. So this is a very exciting finding, and we look forward to advancing into the clinic in this type of cancer. To speak for a moment about our ongoing clinical work, we are doing a multi-phase single study approach to achieve a rapid path to approval. The way this works is on the left, you see part 1 is dose escalation. We've completed the dose escalation work. We tested 3 different regimens, weekly administration, every other week, and every 3 weeks. And then we escalated within each of those dose regimens. And what we saw was the every 2-week administration gave the overall best results. We were able to give higher doses, drive more drug into the tumor, while at the same time achieving better overall tolerability. So we have a really robust data set now on the dose escalation work, and we've advanced to the right-hand side of this slide where we're doing part 2, the dose expansion cohorts. And we've started already the top 3 cohorts, a cohort of ovarian cancer patients with high levels of expression. And the reason we're doing that is because Elahere is on the market indicated for ovarian cancer with high expression. We want to have a data set that we can compare to their data set and determine if we want to go ahead with a head-to-head clinical trial to prove superiority or not. So we need to generate that data to inform that decision. The second cohort is ovarian cancer patients with moderate and low levels of antigen expression. Now, the antibody drug conjugate didn't work in this patient population, and our preclinical data, as well as our dose ranging data, suggests that we will have good effectiveness in ovarian moderate and low, and that may open the door to an accelerated pathway to approval. The third patient population we're exploring is endometrial cancer patients with any folate receptor alpha expression. Endometrial cancer patients tend to have folate receptor levels of expression that are low and moderate, and the ADCs have, at this point, not pursued the endometrial cancer population. We think that we can succeed there, again, based on data that we saw in the dose ranging study. So we have 3 cohorts underway, 2 of which could result in an accelerated pathway to approval, and 1 may demonstrate the ability to outperform essentially the market leader at this point. The next patient population we want to look at is a population of patients with brain metastases. So this would be either a lung cancer population or a breast cancer population, 2 cancers that frequently metastasize to the brain. And we think it could be a game changer when we show effectiveness in those models. To summarize the data that we collected in the dose ranging work, we saw clinical activity in 3 different tumor types: and ovarian cancer, endometrial cancer, and colorectal cancer. So that was encouraging. We saw activity across all levels of folate receptor alpha expression: high, moderate, low, even very low. We had 3 RECIST PRs in our data cohort. We had about 20 patients that had folate receptor alpha expression in the dose ranging. So we really did see some very good activity. We saw good durability. Our longest treated patient so far, and this patient is still ongoing, has exceeded 14 months of stable disease. And we have 6 patients that have gone past 10 months of therapy. Now, remember, these are phase 1 study patients. They are highly pre-treated patients, and so this kind of durability is substantial. We know that Elahere, the antibody drug conjugate that's on the market, in their dose ranging work, the longest duration that they achieved was 8 months. With 1 patient made it to 8 months, and we have 6 patients that are past 10 months, and some of them are still going. So we're pretty encouraged by that finding. And then in terms of, again, the safety, we've seen none of the off-target toxicities that have been seen with other antibody drug conjugates, including no interstitial lung disease, peripheral neuropathy, cardiac, liver, kidney, ocular toxicities. Full disclosure, our toxicities are hematologic toxicities. Because the CDOT drug conjugates are circulating in the bloodstream, you can't avoid interaction with the marrow. So there are some blood cell reductions seen in some patients. But with the every other week administration, the vast majority of these patients that have reductions in their blood cell counts recover before the next administration. This is a self-resolving toxicity. And in a severe case, obviously the physicians can give growth factors. So it's a manageable toxicity profile that we're seeing so far, and clearly differentiated relative to the ADCs. And across all patients with folate receptor alpha expression, we saw about 90% of them had disease control. So if you have expression, you're probably going to benefit from our therapy. To do a direct comparison of Elucida's ELU-001 versus ImmunoGen's Elahere, they have the access to about 1/3 of ovarian cancer patients. That's because they're restricted to just folate receptor alpha high expressing patients in the ovarian indication. They have a black box warning for ocular toxicity. I believe they had about 40% rate of ocular toxicity, and patients are required to have eye exams prior to treatment. Through the first 8 cycles of therapy. There is some peripheral neuropathy and interstitial lung disease. And despite all that, they had a $5 billion market cap as of June 23rd. So let's compare ELU-001. We believe that we'll be able to address over 75% of the ovarian cancer patients. We think that we'll see effectiveness in low, medium, and high folate receptor alpha expressors. We are developing our drug for ovarian, endometrial, and the pediatric AML forms of cancer. Our toxicity profile, we believe, is self-resolving and manageable, mainly hematologic and some GI events. So we're looking forward to an opportunity to create substantial value, potentially more value than ImmunoGen was able to achieve with their first-in-class ADC. And that's the end of my slideshow.
Great, thank you very much, Gino. So let me just gather some of the questions. Give me a second here. I know you addressed some of these questions, so bear with me as we try to sift through it.
Sure.
but I guess we can start with, I know that you mentioned the safety and the targeting FR alpha, but with regards to safety profile, the absence of the significant toxicities such as interstitial lung disease and peripheral neuropathy, which is notable for ELU-001. How has the company approached the development of ELU-001 to ensure such a positive safety profile?
Well, we knew from that, some of the preclinical data that I showed you before, the biodistribution studies, where we just weren't seeing any accumulation in any healthy tissues. We were very excited by that when it was just laboratory information. And then we followed that up with, PK and biodistribution studies in humans, and we saw the same thing. So we anticipated that once we started treating patients and that they were treated over, longer periods of time, that we were going to see a profile emerge that would be different from the ADCs and frankly from any free drug that's administered to a patient that tends to get into, multiple tissues in the body. So we were paid very close attention during the dose ranging work to these particular toxicities. We had requirements in our protocol for deep assessment of these types of toxicities, so that we could really confirm whether they occurred or not. And so far, with over 35 patients treated, we haven't seen anything of concern in any of these tissues. So we're just going to continue down the path of treating these expansion cohorts. We can really explore the efficacy more completely in the expansion cohorts, but obviously we'll collect all the safety data as well within those patient populations. And it's great that we have a differentiated safety profile, and as long as the efficacy comes through at least as good or better than the competitors, then we'll be the market leader, I believe. But you're not going to succeed just on better toxicity profile. You have to have, obviously, the efficacy for this devastating disease.
Of course. I guess digging a little bit more on targeting FR alpha. It's overexpressed in various tumors, but minimally on normal tissues. So how does this selective expression influence the design and expected outcomes for ELU-001?
Well, there's two sources of toxicity with drugs like this. One is, do these antigens express on healthy cells, and does your drug interact with the antigen on healthy cells? That tends not to be the case with folate receptor alpha. The FR alpha expression on normal tissues and normal cells is conserved and not associated with availability of the drug from the bloodstream. So, on target off tumor activity does not seem to be a concern. So the other source of toxicity is just the payload that sometimes can be cleaved and payload and circulate as a free drug in the bloodstream and cause off-target toxicities. We have a very good linker that's highly stable in circulation, so we don't see toxicity associated with free drug in the circulation, and we don't see toxicity associated with off-target targeted types of toxicity. So we feel confident that we can continue to go to patients with lower and lower levels of antigen expression and target the tumors and avoid interaction with the healthy tissues.
Great, thank you. A few more challenges. So I guess every innovative approach comes with a set of challenges. What has been the most significant challenges in developing ELU-001, and how has Elucida overcome them?
Yeah. Well, I have to say, after spending, 35 years in big companies like Pfizer and Johnson & Johnson and Wyeth, one of the biggest challenges I experience is trying to get all this work done with a really small, albeit very talented group of people. You really have to be nimble, and you have to remain extraordinarily focused. I think I shared with you in one of my slides the grand vision that we have for this technology. It involves the development of a plethora of CDOT drug conjugates with different targeting moieties and different drug payloads. And then there's the radiotherapeutics that are a whole nother kind of class of drugs that we could develop for patients. And then there's the immunotherapy opportunity. So, one of the most challenging things in our company is staying focused because everybody wants to, get into some of these other areas as quickly as possible. And I share that desire. But as a really small company that has a small team and, it feels like we're relatively, cash starved sometimes. The goal has got to be to demonstrate that the platform can deliver on all of the promise that we saw in the development, and it's been really a challenge, but something that I'm also proud of. That we've taken this compound all the way to these now patient expansion cohorts in a relatively short period of time. We're generating the data that we need to. It takes discipline, it takes focus, it takes determination, and it really takes all of the effort that our great team is able to put into this development. And again, I'm very proud of the team and proud of what we've been able to achieve so far.
Great, thank you. Let's talk about strategic collaborations and cross-border partnerships. How has Elucida used these types of collaborative approaches to contribute to the advancement of your CDC platform and its potential applications?
Yeah, well, we have a couple different collaborations underway right now. I think I mentioned in the presentation we have one with a top 10 pharma company. And I would say maybe number one, if not, they're probably number two in the ADC field. They came to us. They became aware of our technology, and they said, "Wow, we think that could be next generation technology. We'd like to work with you. We have some targets of interest. We have a number of different drug molecules we would like to try." And it was interesting because the first drug molecule they wanted us to try was a molecule that has an interesting mechanism of action, but its potency just wasn't quite there as a free drug. And they thought, boy, if we can load up your particles with this drug, get a bunch more of it to the tumor, maybe it'll give us the activity that we're looking for. And so we tested that, and we found that it did, in fact, well outperform the free drug in and of itself. Then they asked us to test another drug that was very, very active anti-tumor, had very active anti-tumor activity, but had an off-target toxicity that limited its utility. And they thought, well, if you can avoid uptake into this tissue, for example, we may be able to reduce the toxicity and improve the overall profile of this drug or therapeutic index of this drug. So we're in the process of testing that. We've done some animal biodistribution studies, and we found already that there is limited exposure to the organ of interest here. So there's an early indicator that we may be able to avoid the toxicity that's limited the utility of this particular drug. So there's a couple of examples of how we're working with a pharma company to test different kinds of drugs and different mechanisms for treating cancer, and that collaboration continues today. We've had other companies come to us and say, look, we have a huge market, multi-billion-dollar antibody drug conjugate, but there's now more and more competition, more and more companies pursuing the same target. We need to plan for the future, and if your technology can actually target this target that we like so well, but give us an overall better outcomes, then we can prepare ourselves for the future. So that's another example of how we could potentially work with a strategic to develop a therapy that they need. There have been a number of companies interested in ELU-001 because folate receptor alpha has now been recognized as a validated target. The market is substantial. And we're open to considering various transaction types with ELU-001 that could enhance the development of our company and continue the clinical development of ELU-001. But then beyond that, there are companies that have different types of molecules that bind to antigens on tumor cells with different types of characteristics. There's companies that have molecules called aptamers and DARPins and different types of peptides that are effective at targeting cancer cells. And they've, in many cases, they've made drug conjugates. But when you make a drug conjugate with just a binder, you get one drug per binder. So it really behaves a lot like just a drug in and of itself. But when you can use that binder to target a tumor cell effectively, and you can deliver more drug, you're likely to get a better outcome. So we have two partnerships underway with companies that have components that can be utilized using our particles as well. So again, there's a whole host of different opportunities to develop compounds internally, as well as to collaborate with others in the field to continue to push the envelope and make better and better compounds.
Great, thank you. Have you shared the name of the company, of the big pharma company?
We have not, unfortunately. We're required to. Yeah.
Thank you for asking.
The idea, just to follow up on that, though, the idea is. Sorry.
No worries. I was just curious if you publicly disclosed who that pharma company was.
No, we haven't. But the idea is that once we generate a CDC that they're interested in taking forward into the clinic, then the research collaboration transitions into a typical pharma licensing deal with upfronts, royalties, and milestones. That's what we're aiming for. That's what we're working towards. Yeah. Great.
Excellent. So I guess going more, geographically, right? In terms of global regional expansion goals. Geographically speaking, what is your strategy, priorities there? Are you open to enter the China market?
We are, at the moment, as I said, there have been some companies interested in ELU-001, including some Chinese companies. So we have considered, and we will continue to consider China deals, Asia deals, for example. We're just trying to keep the advancement of ELU-001 going because, I believe that once we demonstrate what the platform is capable of, it becomes a lot easier to sell that we can do it again with another target or another payload. This toxicity benefit or advantage that our CDOT drug conjugates have is related to the particles and related to the construct of our CDCs. So, if we demonstrate we can avoid interstitial lung disease and peripheral neuropathy and ocular toxicities with ELU-001, it's a natural to believe that that's going to occur again when you use a different target or a different payload. So our highest priority is to continue the development, continue to generate a robust data set of clinical data to prove that the platform delivers what we believe it will deliver. And then we think we're going to be very well positioned for, strategic deals as well as, value creation for the company. But yeah, we are certainly open to regional transactions. A China-only deal would be fine. There are some very large global pharma companies, I'd say half a dozen, that are kind of staying very close to us and monitoring the development of ELU-001. And so, I want to try to keep the cards close to the vest, if you will. But there's a lot of interest and there's a lot of activity that happens every day.
Well, thank you very much for your knowledge sharing of Elucida's novel approach, first-in-class target cancer therapy. Very exciting platform with your differentiating novel CDC approach. We're congratulations on all the milestone achievements, and we keep and we're looking forward, being fans from afar, in terms of your accomplishments up and coming. So thank you very much for participating today.
Great. Yeah, thank you. I appreciate the opportunity and look forward to the future.
Great. Thank you very much. This concludes our webinar with me, Mr. Gino Germano from Elucida Oncology for MSQ CEO webinar series. Thank you very much, everyone, for participating. Have a great day. Have a great evening.